Every day within the human body, tens of thousands of DNA-damaging lesions occur. To protect themselves from DNA damage, cells have developed a sophisticated interwoven DNA repair system. This repair system is called DNA damage response, or DDR. DDR is a collective term used for the many different intra- and intercellular signaling events and enzyme activities that serve to detect DNA lesions, signal their presence, and promote their repair. In many cancer cells, one or several of the DDR pathways are mutated, which is one of the mechanisms that enable the cancerous cells to grow and survive, and thus drive tumor development and potential resistance to cancer therapies. Inhibition of DDR may limit the ability for cancer cells to escape. In cancer, several aspects of the DDR differ from normal cells, making it a potential target for cancer treatment. For example, in many cancers there is a loss of one or more DDR pathways. This means the cancer cells are often more dependent on specific functional pathways than normal cells are. These defects in one or more DDR pathways in cancer cells increase reliance on the remaining functional pathways to survive, and may represent an Achilles heel that DDR therapies may be able to target. This is called synthetic lethality. DDR inhibitors are being studied to see if they may have single-agent cytotoxicity in such cancers with defects in DNA repair pathways, as well as in those with high levels of DNA damage and replicative stress. Another aspect in which DDR differs in normal cells and cancer cells is that in cancer, aberrant cell proliferation elicits DNA replication stress and ongoing DNA damage formation, which activates the DDR signaling pathway. Therefore, it is thought that DDR inhibitors may be used in combination with targeted agents, such as other DDR inhibitors that are designed to induce high levels of replicative stress. The possible synergy between DDR inhibitors is being studied to determine if they can serve to maximize DNA damage. A third potential differentiating aspect of DDR in cancer deals with dysregulation or upregulation. Unlike in healthy cells, cancer DDR response is often upregulated. Many of the current cancer treatments are designed to work by causing DNA damage that exceeds the repair capacity of the cancer cell's DDR system. Many of these treatments may cause double-strand DNA breaks in cells. Double-strand breaks are the most lethal type of DNA damage and activate DDR signaling, which enables DNA repair. This can limit the effectiveness of DNA-damaging anti-cancer therapies, such as chemotherapy and radiation, resulting in poor patient outcomes. Inhibition of the DDR pathway is being investigated to establish if it can reduce resistance to DNA-damaging anti-cancer treatment, such as chemotherapy and radiation. This may potentiate the cytotoxic effects of these agents in cancer cells. Finally, DDR inhibitors in combination with immunotherapies are being studied to determine if they promote immunogenicity in a number of ways. One way this may be done is by expanding the neoantigen repertoire. The genetic alterations accumulated by cancer cells create tumor neoantigens. The additional mutational load can potentially increase the immunogenicity of cancers and subsequent responses to immunotherapy. Another way may be by inducing immunogenic cell death, whereby the release of soluble mediators from the dying cells recruit immune cells. Immunogenic cell death can also release cytokines that stimulate immune response. Whether it is increased mutational load or immunogenic cell death, the combination of DDR and immunotherapy agents are being explored to determine its potential to broaden and deepen cancer patient responses. Merck KGaA, Darmstadt, Germany, is conducting clinical trials to identify new approaches to treating cancer using DDR inhibitors. The goal of this research is to find ways to provide additional treatment options for patients.